Welcome to Innovation Day 2023, presented by Connect. We've got such an incredible array of things happening here, supporting the life sci and tech communities in San Diego.
Next up is Dr. Rob Knight, the founding director of the UC San Diego Center for Microbiome Innovation. And if I may say so, he's probably one of the most recognized names in his field. Rob was honored with the 2019 NIH Director's Pioneer Award for his microbiome research and received a 2017 Mastery Prize, often considered a predictor of Nobel. He will tell you why it's important to follow your gut in his talk titled Your Microbiome, the Missing Link Between Your Food and Your Mood. So take it away, Rob.
Okay, thanks, Palin, for that very kind introduction. It was wonderful to see Alison and Kat's talk just now, and some of this follows naturally from what they were telling you about how our gene expression changes under different considerations. Got some disclosures, none are particularly relevant to this talk, but I have to tell you about the cool companies I work with, the UCSD policy. So the idea that we are what we eat is certainly not a new one, and the Italian painter Arcimboldo painted this back in 1591, expressing this idea. The philosopher Feuerbach back in 1863 said, "Der Mensch ist, was ist," the man is what he eats, the first pithy representation of this concept. And about 8 years ago now, I spoke at an event hosted by Jeff Bland, who founded the Personalized Medicine Lifestyle Institute. And he has this quote I'm particularly struck by: "Food is a language that speaks to our genes." So what he means by that is that our genes are fixed when we're conceived, but what they do depends tremendously on what we eat. And I'm sure you've all heard this admonition that you should eat the rainbow, that when you were at the buffet there, you should be picking out a range of brightly colored fruits and vegetables. And that's true, and we're learning more and more about how the language of food is expressed as a language of color. So everything from the carotenoids that make the carrots orange to the anthocyanins that make the berries purple are chemical compounds that have an impact on our gene expression and on our gut. And it's interesting to think about this in our daily life. And regrettably, the source of food closest to my house in Pacific Beach is this one. I take my kid there. She sees a lot of bright colors. What I think is especially ironic is that their motto is "too much good stuff," whereas you might suspect that they've stripped out all of the good stuff and replaced it with synthetic analogs that have completely different physiological properties. And this concept of ultra-processed foods in particular is more and more of concern. So I put a couple of quotes from this article that came out last year, where physicians quoted in the article said things like, "Health consequences of ultra-processed foods are dire. They can lead to things like type 2 diabetes, cardiovascular disease, dementia, all sorts of other things that you don't want." So this makes me wonder what the astronauts were eating up in the ISS. Unfortunately, we don't have a lot of good records of their diet, although we've been doing other stuff looking at the habitat there. Fortunately, last year, a solution was found. Apparently, according to the news reports, cheese protects you from all causes of death, says science. And why would science say it if it weren't true, right? So this is great, right? It implies that humanity struggle with the Grim Reaper. You can fight him off with cheese. You're done. Simple solution. Of course, it's not quite that simple, and the problem is that large-scale studies of diet are really challenging. And this study from a group at the Harvard School of Public Health back in 2011 is still one of the largest of its type. And they looked at 120,000 people over 20 years, looking at the impact of each food item on weight loss and weight gain over that period. And I've got to say that despite all of this heroic effort, what they found complies, I bet, with exactly your stereotypes of what thin people eat and what fat people eat. So if we look at the single food most associated with weight loss, what it was yogurt, perhaps no surprises there. If we think about the food most associated with weight gain over this 20-year period, what it was potato chips. Again, perhaps not much of a surprise. But what surprised me was the size of these effects. So each serving of yogurt per day led to a weight loss of about 372 grams over 4 years, whereas each serving of potato chips a day led to a weight gain of about 3/4 of a kilo over those same 4 years. So think about what this means. If you're perfectly average for this 120,000 person population, what it means is that you have to make the right choice 1,460 times in a row, giving up the chips you were going to eat and eating a serving of yogurt instead, and it's only going to change your weight by a couple of pounds. So that's maybe not very impressive, right? But what this is masking is individual variation. So these are population average data, but every individual is different. So some people are going to make that change, they'll lose 20 kilos. Other people will make the change, they'll gain 19. It all averages out when you look at the whole population. So we've spent an enormous amount of time looking for the key to these links between diet and health in the human genome. And this search has cost billions of dollars. There's this entire field of nutrigenomics where the idea is that you're going to analyze your human DNA, figure out what you should eat based on your differences in the genome compared to other people around you. And what I wonder is all of this research like the proverbial drunk man looking under the streetlight for his keys, not because that's where he dropped them, but because that's where the light is. And what I wonder is, are we looking in the right place when we're looking for genes associated with traits linked to dietary response, or are the keys somewhere completely different? Now, what I'm talking about is what we see when we consider where are the genes in the human body. So the human genome has about 20,000 human genes. This is what was mapped in the Human Genome Project. But if we look at the size of our microbiome gene catalog, that's more like 2 to 20 million microbial genes. And so by that measure, you could say that we're only 1% human in terms of our unique genes. But what's more important is that the 99% of our genes that we ignore when we focus solely on the human genome, those are the 99% that we can change. They're not fixed at conception. We change them through our lives, through all sorts of things that we're doing. And it's urgent that we study all of this complexity because we're losing a lot of it. So when we look at people like the Hadza hunter-gatherers in the Great Rift Valley in Tanzania, the birthplace of humanity, what we find when we look at their microbiomes is a tremendous amount of seasonal cycling. But we also see that when we look at populations around the world, they sort out automatically from the least industrialized, like the Hadza, to the most industrialized, like us, the Australians, the Japanese. And this is coupled to a tremendous loss of microbiome diversity. So all of these major kinds of microbes that we've co-evolved with as a species and as a lineage for millions of years, we're losing these major groups and replacing them with other completely different and less diverse kinds of microbiomes. So it's almost as though we took the rich inner rainforest of our gut and we bulldozed it and we replaced it with a cityscape where only the pigeons and the rats survive. So I'm sure you're all familiar with Rachel Carson's work from the 1960s, where she documented how the use of DDT and other pesticides was wiping out the bird life around us. My good friend and colleague Marty Blazer, now at Rutgers, has documented something similar for our inner ecosystems in his book Missing Microbes, where he describes how not just antibiotics, but also all kinds of other things, including low-fiber diets, increased use of C-section, decreased breastfeeding, are depleting our microbiomes right now in ways that we could reverse. And Marty likes to show this pair of graphs, which just amazed me the first time I saw them. So what you see in the first graph is how infectious diseases caused by one organism were brought under control during the 20th century. But at the same time, we see this tremendous rise in so-called non-communicable diseases, so multiple sclerosis, Crohn's disease, type 1 diabetes, asthma, all skyrocketing. And 20 years ago, when this was published in the New England Journal, we didn't know any of those diseases had anything to do with the microbiome, whereas today, all of them have been linked to the microbiome, not just in animals, but in humans as well. And so we now know that the gut microbiome provides an incredibly sensitive record of all kinds of things, including how we're born, how we're fed early and later in life, what diseases we have and how we treat them, even things like how much exercise we get. Now, I've talked mostly about the gut microbiome here, but it's important to remember that the gut is not Vegas, and that what happens in the gut does not stay in the gut. And so stuff that happens in your gut microbiome affects your whole body, including your immune system, including your brain, including pathogen clearance. So it's much broader than just digestion. And then the negative aspects, the gut microbiome is also linked to a wide range of different diseases, including kidney disease, other metabolic disorders, GI disorders, cancer, and even increasingly disorders of the brain. So we're increasingly turning to machine learning to understand this individual variability. And one of the first efforts in this respect came from the Weizmann Institute, where they were able to show that when you take 800 people and you hook them up to continuous glucose monitors, so you can look at the impact of each food item on your blood glucose response, feeding people a defined sequence of diets. What they found was amazing, because when they averaged the glycemic index for each food across those 800 people, they pretty much captured the published values exactly. But when they calculated individual glycemic indexes for each food for each person, they were completely different one person to another. So for example, tomatoes are healthy for most people, but one of the people they looked at here, their blood glucose spiked every time they ate tomatoes. When they cut them out of their diet, they did much better. And one of the key findings of all this was that most of us think of rice as a pretty healthy food, right? But for a sizable fraction of their subjects, it was actually worse for their blood glucose to eat rice than it was to eat ice cream. And the deciding factor was what microbiome people had, right? So on learning this, people typically have two questions, right? The first question is, am I in the rice category or the ice cream category? Is there a test I could do that could diagnose that from the microbiome? Sure enough, there is. But more interestingly, suppose I do this test and I find out I'm in the category of people who should eat rice. Could I get myself into the category of people who should eat ice cream instead? Which is a very interesting question. May seem a bit frivolous in this context, but in the case of cancer, it's deadly serious. And I'll come back to that at the end of the talk. So between this work and work that's been going on in my lab since 2011, when we introduced machine learning to the microbiome field, we can now build predictive models for a lot of different traits related to the microbiome. And one thing I've been working on for the last 15 years is obesity. And today, for example, I can tell you with about 90% accuracy if you're lean or obese based solely on the genes and the microbes in your gut. So on the one hand, we think this is an awesome trick from a technical perspective. On the other hand, we don't think it's got a lot of commercial potential as a test for obesity, right? Because I bet you can tell which of these people is obese without doing any sequencing at all. But if you try to do that classification task, lean or obese, based on the human genome, you can only do it with 57% accuracy, whereas we can do it with 90% accuracy for microbial genes. But all this is association. So you might wonder, do people become fat because their microbial genes make them fat? Or do they become fat and then the microbes respond to that obesity and change afterwards? And we really need to get at causality to understand how this works and to design interventions. And so to do this, we teamed up with Jeff Gordon back in 2005 at Washington University. And with the technology that he really pioneered there of gnotobiotic mice, which we now have set up here as well at UC San Diego, what we can do is we can raise mice in a bubble with no microbes of their own, and then colonize them with the microbes that we think are going to have an impact on their bodies. And so in the case of obesity, you can do this even across the species barrier. So you can take microbes from an obese person, put them into mice, and the mice will gain weight much faster than if you instead give them the microbes from a lean person. So this doesn't necessarily establish causality in humans, but it does show that in a mammal, we can change their weight by changing their microbiome. And this has been done now not just for obesity, but for a whole lot of other things like colon cancer, with Sarcus masmanian. We've done it for Parkinson's disease, for multiple sclerosis, for autism, and now even for Alzheimer's. Although working out the specific pathways takes a lot of additional time. So think about these results. We can take individual differences in people and transplant them all the way into animals, into a completely different species, by transplanting the microbiome. That's pretty cool, right? And so I'm learning this. A lot of people have asked me, do I think that fecal transplant is going to be a cure for obesity? And just to give you a bit of background on fecal transplant, this is the map of different microbiomes that we produced in the Human Microbiome Project, which the J. Craig Venter Institute, among a number of other institutes, was involved in doing the sequencing for. And what we did together is we produced this map of how microbiomes are arranged. We're emerging like different continents on this map. You see the mouth, the skin, the vaginal, the fecal microbiomes as different areas. And I'm going to show you fecal microbiomes from people with C. diff. That's those orange spikes, and you can see they're in a completely different part of the map from that healthy region down the bottom in brown. And what's going to happen is that 4 of these patients, and this is work we did with Alex Kreutz and Mike Sadowski at the University of Minnesota, 4 of those people are going to get a fecal transplant from one healthy donor. And if you're wondering what fecal transplant is, this is Bill Sanborn, who used to be our chief of GI at UC San Diego, about to do his first one with a huge grin on his face, and also with a GMP and GLP prep of fecal material from a nonprofit called OpenBiome, because the FDA does regulate stool as a drug, so don't try this at home. But anyway, 4 of these people had had diarrhea for years to be eligible for this trial and had failed antibiotic therapy, get a fecal transplant from that healthy donor in the healthy range at the bottom, and each frame in this animation is their microbiome journey after that fecal transplant. And what you can see when I start this going is immediately their microbiome ecology completely resets into the healthy state, and you can see they stay in that state for months of follow-up, and this is coupled to complete remission of their clinical symptoms. And in general, fecal transplant has been 90 to 95% effective for C. diff in different studies. Antibiotics are only 30% effective, so it's rapidly becoming a first-line therapy around the world. I was just in Australia talking about this, where their regulatory agency has recommended that it's the frontline therapy for recurrent C. diff. And so the question that faces us is, for what other diseases can we identify a problem with the microbiome like this, and then provide guidance on how to bring it back into health for all of these different diseases that we've now linked to the microbiome, including things that you'd expect, like inflammatory bowel disease and IBS, things that are more surprising, like cardiovascular disease, rheumatoid arthritis, and then things that are really surprising, like Parkinson's disease, autism, other things related to the gut-brain axis. And so we need to find the good and the bad places on this map and understand how to move you around on it, whether it's something that seems as extreme as fecal transplant, or something that seems as gentle as diet, which I'll say more about later, or something in between, including phage therapy, which we're doing at UCSD via IPATH, advanced probiotics, prebiotics, and so forth. So this is all very impressive for the specific case of C. diff, but you might be worried about whether there are limits to what fecal transplant can achieve. And I get asked about this so frequently that I realize that I have to temper people's enthusiasm. And so I like to tell a story about a dog who I had in grad school, and she really envied the geese that could fly up above her, and she'd spend hours and hours watching the geese, presumably because she also had the power of flight. And at some point, I think she realized, I know, what I'm going to do is I'm trying to get this phenotype and transfer it from this other species to myself via fecal transplant, which she did very enthusiastically via the route available to her. But sadly, despite doing that over and over again, this only happened in her dreams, right? So so she was not able to get that phenotype, and there's a lot of other phenotypes that we think will not be transferable either. So we do have to be responsible about what we promise, and the clinical trials for obesity for fecal transplant in humans have all failed, although other indications like IBD and, interestingly, autism have had a remarkable amount of success to date. So besides fecal transplant, though, there are a lot of other ways to change your gut microbiome. And in the American Gut Project, the citizen science project that I run at UC San Diego, which is now part of the larger MicroZeta initiative, what we've been doing is we've been looking across a lot of different people and a lot of conditions to understand what makes a difference in your microbiome. And one thing that was fascinating to us is that one of the largest impacts on your gut microbiome is how many different species of plants you eat. So this is something that's completely under your control. You don't have to chow through a giant bucket of kale at every meal. If you eat a lot of different kinds of plants, that actually has more of an impact than whether you have inflammatory bowel disease, more of an impact than if you're on antibiotics, and way more impact than things like how fat you are, how much you drink, all kinds of other things that we can also see impact the microbiome when we go to larger sample sizes. Now, one thing I mentioned in the context of diet was, wouldn't it be great if you could find out that you were in one group versus another in terms of what you should eat? And I should note that this kind of thing is already saving lives right now in the context of cancer immunotherapy. So Jen Wago at MD Anderson has been a pioneer for this with checkpoint inhibitor therapy, so Keytruda, If you have melanoma, Keytruda works amazingly for you if you're in a group of about 30% of people, but for the other 70%, it basically does nothing. And so, a few years ago, Jen found that the difference between the responders and the non-responders was their microbiome. And what she's been doing is she's been using fiber and probiotic interventions to identify the people who are not going to respond to Keytruda and change their microbiome so that they respond to it and so that they live. And so the fiber interventions have been remarkably successful, but I do want to highlight that the probiotics actually made the cancer patients die faster. And using that technique of gnotobiotic mice, where you start them germ-free, you give them the human tumor cells, and you also give them the microbiome of the individuals. If you give them probiotics as well, for 2 different off-the-shelf probiotics, they also died faster. So we have to be really careful about taking conclusions from healthy people and applying them in disease populations. We have to redo the clinical research in a careful and measured way. But the real problem, whether we're talking about this in a clinical setting or whether we're talking about it just for our own daily lives, is that we need to build a better user interface to our microbiomes. And as those of you who've had microbiome profiles done, what you get right now is you get a list of 1,000 species or 1 million genes or other very intractable data full of Latin and Greek words that unless you're really a specialist, you don't understand. And that is not going to help you picking out a particular product in the supermarket. So what we're working on right now is a big new idea at UC San Diego, is natural language interpretation and interrogation of microbiomes. So the idea is that while you're standing there in the supermarket, I'm sure you've all heard of ChatGPT, right? We're calling this crap GPT, where the idea is you should be able to ask your phone in natural language questions like, should I eat this particular product to improve my health in different respects? Or more ambitiously, maybe you could have your phone highlight on the shelf using the camera and augmented reality the things that are good or bad for you personally based on your genome and your microbiome, rather than just generic recommendations like the Mediterranean diet. So that's the kind of crazy stuff we're up to at UC San Diego and the Center for Microbiome Innovation, with over 140 faculty members who I'm not going to thank individually, even a large number of collaborators, awesome people in my lab, many different sources of funding that we're very grateful for, including the tens of thousands of you who have participated in American Gut. And finally, thanks for having me here. I'll be delighted to answer some questions. Thank you.
I really feel passionate about sharing the San Diego talent, elevating San Diego, because it is the best place to be an entrepreneur.